Hello guys, welcome to my channel. So today's topic is what are transgenic animals and how they are created. So the transgenic animal, it refers to an organism containing integrated sequences of cloned DNA which is transferred using techniques of genetically engineered, which are known as transgenic animals. So the transgenic animals produce with the purpose of producing better and good quality breeds. Nowadays there are a number of methods which are available to produce transgenic animals like pronuclear microinjections, embryonic stem cell manipulation, etc. And they have contributed in many ways to the agriculture, medicine, food, disease models, etc. There are various benefits and risks in these, that maybe a fewer food supplements and offsprings could be established in one generation. The risks can be as insertion of foreign gene may upset the expression of genome and normal reproduction. There are various other methods of transgenic animal production such as pronuclear microinjection, embryonic stem cell manipulation, viral vector cytoplasmic injections, nuclear transfer, primordial germ cells, etc. There have been many trials on animals using green fluorescent protein. A dog breeder called David, in the Netflix show Shown, had used the puppies to make them fluorescent, but it would take some years and it is not actually possible to do that. So the production of a transgenic animal can be done by a human antithrombin gene, which can be taken through microinjection into a egg of a donor goat, and it could be developed into a host goat, and that turns into a transgenic goat, which gives us the antithrombin which would be present in them. The goal of genetic engineering is to create a transgenic organism. So what a transgenic organism is, it is an organism which has genes from two different places. It has genes from itself that is got from its parents, and it also has genes that were put into it by a scientist. So there are 3 steps to create a transgenic organism. The first one is that we are going to cut a gene from an organism that we are taking it from. So in the example, we are giving a human large hands. We would find an organism that has really large hands or wings and cut that gene from large hands or wings out of that organism. Then we're going to paste it into a piece of DNA called a plasmid. Over here on the side, the circular piece of DNA is a plasmid, and it's got a couple of different important sections that are needed in order to paste. Then the last step is to deliver that plasmid into a second organism. So taking this little circular piece of DNA that contains your gene for large hands and putting that into a human. So this plasmid, the circular piece of DNA over here on the side, contains two very important parts. The first one is the gene of interest, or the gene that we are trying to put into the new organism. So in this case, the genes for really large hands. And the second one is a genetic marker. A genetic marker identifies plasmids that contain the gene of interest. The lot of times a marker is antibiotic resistant, so that we can grow it on a plate of antibiotics. So it kills the ones without the plasmid and it will keep the ones with plasmid. So the step 1 cutting, a scientist cuts DNA by a special molecule called a restriction enzyme. So a restriction enzyme is a chemically essential that cuts the DNA in a very specific place. So if we had, see, we have got 2 molecules here of DNA that we can see that are double stranded, and the C's are bound to G's and the A's are bound to T's. the enzyme comes along and finds a very specific sequence. In this case, it finds the sequence ETAG, and when it sees the sequence, it cuts the DNA, and it cuts the part of the single stranded part over here. So we're cutting two different pieces of DNA with the same restriction enzyme, and because the restriction enzyme found the particular sequence in both molecules, it's going to cut them both. And so these single stranded pieces of DNA on the end, they are called the sticky ends, and the reason that they are called so is because they are able to bind together. Now we are able to see here the G is complementary to the C, so those two can bind together, and the A binds to the T, and T binds to the A, and the C binds to the G. So these sticky ends allow two different pieces of DNA to stick together. Now the step 2, it's pasting our DNA on using the sticky ends that we have created while we cut the DNA. So pasting this little piece of DNA into our plasmid, we have got the sticky ends on each side. So this is sort of like molecule A and molecule B from the last slide, and now we're going to paste this gene that we want to put into our new organism into the plasmid. So this is like our gene for big hands, and we are going to put it into a circular piece of DNA, and it's able to fit in there because of the sticky ends or the single stranded parts that we have created by cutting both of these with the restriction enzymes. We have explained already what the plasmid is. Okay. Step 3 is delivering the DNA to the desired organism. So we're going to take this gene for large hands and deliver it to the humans so that the humans have really large hands so that they can fly. But the first thing that we want to do when delivering it to a desired organism is to make a lot of copies of this little plasmid, because humans have a lot of cells and we want to infect all of them. So to make the copy of a DNA gene, we have to make a copy of the plasmid, like what we can do with it, and put the plasmid into the bacteria. And the process of putting the gene into the bacteria is called transformation. So we put the plasmid into the bacteria, and the bacteria can make copies of themselves or reproduce really fast. So they reproduce using asexual reproduction. So they are making exact copies of themselves, and as the bacteria makes an exact copy of itself, it also creates the exact copy of the plasmid that we stuck into it through the process of transformation. So here's the genetic marker comes in, and why it is so important to the genetic marker we said we are going to use is resistant to the antibiotic. So we take those bacteria and we grow them on this plate that's full of antibiotics. So if the bacteria is resistant to the antibiotic, those bacteria are going to live. And so if we have got a lot of bacteria that's alive on this plate, we know that our gene has made it into the plasmid. So then we are going to take these bacteria and we are going to infect whatever organism we are trying to put the gene into. So we are going to infect a human here with these bacteria so that they will deliver this gene for large hands to the humans. Now if the bacteria doesn't survive, it's because they didn't get the resistance gene and they didn't get the gene for large hands. So we know that these bacteria are useless. They didn't then in work essentially alright. So now we are going to take the bacteria and infect the desired organism. So this is kind of a summary like what we did. Yeah. Okay, so we took a bacteria that has a plasmid, a circular piece of DNA, and we cut the plasmid. We also cut the gene out that we want to put into our new organism. So here's the gene for large hands that we created sticky ends by cutting them with the restriction enzyme so that these two were able to stick together. And now we have taken the gene for large hands and we put it into a circular piece of DNA through the process of transformation. We took this circular piece of DNA and put it in the bacteria. This bacteria is going to reproduce and make lots and lots of copies of itself. So here is the bacteria, and now we are going to take this bacteria and we are going to take this bacteria and make sure that we have actually transformed these bacteria, and we are going to grow them on plates and we are going to grow them with antibiotic. If they survive, we know that the gene is there, and we are going to take all the surviving bacteria and infect the plant or the animal we want to put the gene into. And now we have humans with large hands. So here is another picture of exactly the same thing, and it's a little bit larger and more detailed than the last picture. And you can review this picture. Can write your doubts in the comment section below. Thank you. And you can understand it in a better way, and you can even explain it to us in the comment section, like how to create a transgenic organism.